Welcome to the B191 video lecture on DNA fingerprinting and molecular systematics. Thanks again to Jennifer Crossman for some beautiful slides. The study of DNA is one of the most fascinating topics in biology, and here are some cool facts to set the table. Did you know that there are 3 billion nucleotides in a single human cell? Of those 3 billion, about 0.1% is unique to an individual. With this level of uniqueness, there are lots of ways we can use DNA. DNA can be used to identify individuals, either in the aspect of who committed a crime or identifying human remains after a catastrophe. We can determine paternity, and where would daytime television be without paternity cases, I ask you? We can determine who is likely to carry a disease or what the prevalence of disease is in some society or culture or country or region, and we can study how closely related are different species of animals. For example, recent work shows that falcons are more closely related to parrots than they are to hawks and eagles and owls. Who would have thought that? Before we can use DNA, we have to know how to gather it and how to use it. Let's look at some terms to help guide us. DNA sequencing refers to the process of determining the sequence of nucleotides in a strand of DNA, the sequence of the C, A, T, C, C, G, G, A, etc. We use this to compare relatedness among species or other taxa, species, genera, families, etc., like the falcon-parrot example. Genotyping, on the other hand, identifies the alleles possessed by an individual. With genotyping, we identify individuals, like in the forensics example, or close relatives, like in the paternity example. Let's examine DNA sequencing first. Again, we compare the exact sequence of nucleotides of two species. We calculate the percent similarity of the sequence, or sequences, and based on the similarity we construct a phenogram, or a tree, to describe the pattern of relatedness that we observed. You're going to be doing this in lab, and here's what it's going to look like. Here you see a partial sequences for two species. Each pair of letters is called a base pair. The bars that connect a pair show that the pair possesses the same nucleotide, and no bar, or the absence of a bar, means that they're different. When you compare sequences for multiple species, you construct a percent similarity table. Here are species B and C. We see are 90% similar in their sequence, but species B and A are only 57% similar. Next, you build a tree based on the similarity table, and if two species share a shortest branch, they are the most similar. So looking at these three choices of the tree, tell yourself which of these trees reflects the table to the left. The best choice is the tree that's on the left of the three trees, because B and C have the shortest branch between them, and A and C are the farthest apart. There are lots of ways to draw trees, or phenograms, to express relationships. Shape is only one factor to consider when drawing a tree. What you really need to know is that different trees are built on different types of information, and therefore they have different names. Pay attention to and remember these two terms, cladogram and phylogeny. This tree is a cladogram. Cladogram is C-L-A-D-O-G-R-A-M, cladogram. And a cladogram expresses relationships in terms of characters or traits shared by the organisms. In the table, all species with a red 1 share a character. They have jaws. Also, the species with the yellow 1, or the yellow box around the 1, share some other character. They all have lungs. We map these traits on a tree, with the species that share the most characters being closer together. The lamprey on the left of the tree is a type of fish that has no jaws. It only has a sucker for a mouth. They go first. All of the other animals have jaws. Sharks have no lungs. They go next. All of the animals to the right of the shark have lungs. Gorillas and humans are similar in all traits except the last one, and they share the shortest branch. Now, here is the crucial point for you to get from this slide. We are mapping physical characters. We could just as easily build a tree for something that is not alive at all, like cookies, right? Has cinnamon? Yes or no? Raisins? Yes or no? This particular tree does not necessarily imply an evolutionary relationship. We are simply talking about shared characters. Once again, this tree does not necessarily imply any sort of evolutionary relationship. This tree is called a phylogeny. This tree does imply an evolutionary relationship. This tree is not built on some physical character. It is built on percent similarity of DNA. DNA is the currency of genetics, heredity, and evolution. This tree expresses a molecular relationship. Here, if two species share the closest branch, they are closest evolutionary relatives. You can see that dog and wolf in this phylogeny are closest relatives, more closely related than foxes or other animals in the tree. A phylogeny even includes a timeline. By seeing how divergent two sequences are, we can calculate how long ago two species diverged from each other. That is the power of DNA technology. Now back to genotyping. Genotyping relies on polymorphisms. Polymorphism simply means many forms. A polymorphism is a variation in a DNA sequence between individuals of the same species. If we use a restriction enzyme to cut a DNA strand at a restriction site, we get fragments, and no two individuals will have the same DNA fragment. The name restriction fragment length polymorphism implies this. An STR is a standard tandem repeat polymorphism. It is a polymorphism that repeats itself in a strand of DNA, and different individuals have different numbers of the repeat. Yours may be repeated X number of times, and mine may be repeated Y number of times. These STRs occur on alleles and are inherited like any other trait. And like any other trait, we know the frequency of these STRs in the population of the United States. U.S. forensics uses 13 different STRs to differentiate DNA samples among individuals. And this can be done because the probability that two individuals share the same 13 STRs is 1 in 7.7 quadrillion. You have a better chance of winning the lottery than being mistaken or have your DNA mistaken for the DNA of some other individual. One of the first steps in genotyping is PCR, polymerase chain reaction. PCR takes a really small sample of DNA from a single drop of blood, or the tissue at the end of a single hair follicle, and replicates the DNA into a larger sample that can be tested. The steps are: DNA double helix strand is denatured into single strands. A primer bonds onto the single strands of DNA, and the strands are copied, and this process is repeated over and over and over until you have created a large sample from a small sample. The second phase of genotyping is electrophoresis. Electrophoresis uses an electric charge to separate fragments of DNA according to their size. DNA is negatively charged. In electrophoresis, DNA is placed into a gel, and a charge is run through the gel. DNA will migrate toward the anode. Because DNA fragments are moving through a solid substance with pores, the gel, Smaller fragments are able to move farther than larger fragments. When you are done, you have a map of fragments that can be read, sometimes under a black light, and compared for similarities. Does this sound complex and out of reach? Well, you get to do this in lab right here in B191. Here is the scenario. A crime has been committed. You are a scientist working in the FBI forensic lab. FBI agents have provided you with a biological sample, hair, from a murder crime scene and samples from 3 suspects. It is your responsibility to identify who was involved in the crime. Steps 1 to 3 of this process have been done for you in the forensics lab already. DNA has been extracted from the samples, amplified using PCR, and cut into fragments with the restriction enzyme. You have to separate the fragments via gel electrophoresis, stain the gel, and compare samples to identify the suspect. That is the end of the video presentation. Thank you very much. Have a good week.